My name is Lynn Nygaard. I'm the director for the Center for Mind, Brain, and Culture, and we are delighted to welcome Professor Simone Shamay-Tsoory to virtual Emory. Professor Shamay-Tsoory is professor of psychology at the University of Haifa in Israel, and that's where she's joining us from. So thank you. Thank you for accommodating sort of a late night talk for you, or at least a late evening talk. She's senior researcher of the Brain Activity Project and directs the Social and Affective Neuroscience Lab. And I'll just give a brief introduction, and obviously Dr. Shamay-Tsoory is going to do a much better job describing her research, but her work seeks to understand the neural mechanisms broadly underlying social behavior, social cognition, and emotional experiences. Her work addresses these fundamental questions, such as how do we understand emotions, and what are the neural underpinnings of constructs like empathy, social communication, and interpersonal synchronization. To address these issues, she uses a variety of neuroscience tools, including neuroimaging, neurostimulation, and psychopharmacological techniques. And her lab works with various populations of patients, as well as with healthy individuals. So her lab is interested in both the cognitive emotional consequences of brain pathologies, whether psychiatric, acquired, or developmental, and examining emotional and cognitive brain functions in healthy subjects using the aforementioned imaging and brain stimulation techniques. Within these broad themes, her lab investigates a variety of topics, including the role of oxytocin in social behavioral, neural underpinnings of social comparison and jealousy, the neural underpinnings of social touch, and chemical social communication using odor. We're delighted to have Dr. Shamay-Tsoory here. She'll be talking, her title of her talk is The Empathetic Brain, or Empathic Brain, the Neural Underpinning of Human Empathy. And I just wanted to say that before we start, we're going to request that you save your conceptual questions for the Q&A period at the end of the talk. But if you have questions of clarification, we ask that you throw them into the chat, and one of us will monitor the chat and ask Dr. Shamay-Tsoory to clarify. So please join me in welcoming her to Emory and to the CMBC. So thank you so much.
Thank you, Lynn, for the kind introduction. Okay. So thank you for inviting me, and thank you for the kind introduction. And I'm really excited to be here, and so many people that that share a similar interest in this work, and I hope that you'll find it interesting. I would like to present to you a few insights from our lab about the neural underpinning of empathy. And basically, I would like to present a new approach that we are trying to work on in the lab that views empathy as a feedback loop, as an interactive behavior between an observer and a suffering target. And first, I would like to thank my collaborators, the funding agencies, and the students that participated in running the experiment. So before we start to examine the neural underpinning of empathy, it's very important to define empathy. And there are many definitions for empathy, some of which are more emotional and deal with the emotional aspect of empathy, like emotional contagion, which is basically our tendency to share the emotions of other people. Other aspects of empathy are more cognitive, like perspective taking, which is basically understanding someone else's emotion and mental state without actual emotion sharing. And it is now believed that empathy involves both aspects, both emotional and cognitive aspects. Empathy occurs in almost everyday situation involving distress, when we share someone else's pain or sadness, but also in positive situation when we share someone else's joy or success. It occurs throughout the lifespan. We see it in young children, we see it in adolescent, indoors, outdoors, almost in every interaction. We experience this emotional sharing and cognitive understanding of someone else's pain. And I have here a short clip demonstrating real life empathic behavior between siblings.
Oh no.
Oh, sorry.
It's okay. You're strong. Do you want carry? All right. Here, Freely. Now I'm gonna carry you.
Yeah.
Okay, so in this very nice example, we see that empathy involved this trial and error behavior. The brother observes his sister suffering, and he's trying one strategy to help her, and it doesn't work, and then he tries another strategy. So it involves this dynamic interactive behavior. And the question is how to examine this interactive behavior in the lab when we want to study the neural underpinning of empathy. And unfortunately, in neuroscience, the vast majority of neuroimaging experiments involve those isolated agents lying in the Fmri scanner. They carry out artificial tasks, mostly computerized tasks in sensory and socially deprived environment. And the social interaction is extremely limited, and it very remotely resembles this dynamic real-life interaction. And there are two fundamental limitations in most traditional experiments in neuroscience of social behavior. One is person-dependent limitation, and the other limitation is situation-dependent limitation. The person-dependent limitation basically revolve around the limitation of activity of the participant. If our participant is unable to move, it may basically interfere with their sense of embodiment, their sense of agency, and it interfere with their basic ability to react as they do in real life face-to-face interaction. The situation-dependent limitation basically focus on the decontextualized environment that we see in most social experiments, social interaction experiment, where we have basic paradigm involving computerized paradigm with no real face-to-face interaction. So if the traditional approach involves an isolated participant carrying out artificial tasks, a new approach in neuroscience suggests that we should have in the lab real face-to-face interaction and examine both brain and behavior in the lab. And the real life hardcore approach suggests that we can even take our experiment outside the lab and measure brain and behavior in real life experimental setting, outdoors with portable equipment, and measure both brain and behavior. So in the context of empathy, the most traditional studies of empathy that examine the neural underpinning of empathy basically used models of an open loop. They examine how an empathizer reacts to the suffering of a target. However, when we look at empathy in real life interaction, having both an empathizer and a target interacting, we can perhaps view empathy as a feedback loop. We can perhaps view how the distress of the target affect the response of the empathizer, which reacts and then affect directly back the target. So we can see how and the empathizer may adapt the empathic response based on the incoming feedback and adapt the response to the target, and then some mutual adaptation can occur. And this is fundamentally different than the traditional one open loop models. So basic models of empathy, traditional models of emotional empathy suggested that brain regions involving the anterior cingulate and the insula mostly react in sharing pain, and the regions like the inferior frontal gyrus and the IPL are more involved in, or the observation execution or mirror neuron system are also activated during emotional empathy. On the other hand, in cognitive empathy, we see other regions like the ventromedial prefrontal cortex, the dorsomedial prefrontal cortex, and these are the traditional network that usually appear in fMRI studies of empathy when we measure the reaction of participant to the distress of other. However, the new two-brain approach suggests that instead of looking at neural activation in one brain, perhaps we should look at two interacting individuals as an extended neural network involving both intra-brain connections and inter-brain connections. So according to this hypothesis, according to this model, the two-brain model approach suggests that social interaction are basically a representation of an extended functional network with intra-brain connection that are probably mediated by physical connection and interbrain connections that are mediated perhaps by sensory input. And we use the term hyperscanning to represent the measuring of brain activity from two or more individuals interacting simultaneously. So when we started to think about using the two-brain approach to study empathy, we looked for an empathic response that may have a direct effect on the target. And we decided to focus on social touch. We know that from the work of Frans de Waal that empathic touch in the context of consolation is a behavior that is associated with reduced distress, and it is associated with empathy, and it has been reported not only in chimpanzees, but also in bonobos, in elephant, in rodents, and across, and of course, across human culture, we can see a consoling touch or comforting touch using in situations that involve empathy. And there are experiments that show that using touch, we can communicate emotion and even identify emotion through touch. So we decided to focus on empathic touch to test this empathy feedback loop. And in the first phase, we wanted to see which type of touch we should use as a comforting touch. So we asked participants to choose between three types of touch. Slow stroking on the arm, which basically activate the CT fibers, and it is considered a pleasurable touch. Slow stroking on the palm and static hand holding. And we found that 75% of our participants selected hand holding as the most preferable touch aimed at reducing their distress. We recently replicated this study in an online survey when we asked participants also to choose which type of touch they would prefer in various emotional and physical situations involving distress. We asked them to imagine that they are with their partner and they are having all sorts of physical distress or emotional distress. And we asked them to choose which type of touch they would prefer. And we found in 4 experiments that the hand holding was significantly more effective in reducing distress, and participants prefer this touch over stroking, both in situations involving physical and emotional distress. Also in study 3, we asked the participant with injection phobia to select the type of touch that they would prefer. And this is in women who had recently gave birth. Also, we asked them which type of touch they wanted to have while having labor, and we see overall a very strong effect of hand holding. So we focused on the effect of hand holding in reducing physical pain. And this is Pavel Goldstein, my former PhD student, who in the first experiment started to examine the analgesic effect of social touch. And basically wanted to examine if hand holding indeed reduces distress, and whether hand holding, the toucher empathy has a moderating effect on the level of analgesia. So in this experiment, we recruited 20 romantic couples, and they were divided into a target and a consoler, or the one that was providing touch. And we had 4 conditions: no touch, partner touch, stranger touch, and alone. The target was inflicted with pain using a thermod attached to the arm. And the partner was instructed to either hold the hand or just be present in the room without any touch. And we can see clearly here from this figure that the level of pain was the lowest in the partner touch condition as compared to the other control condition. And the stranger touch was ineffective, as we see here. So we see here that hand holding is indeed important and contribute to pain reduction, but it is also important to look at the identity of the toucher, because the stranger touch is not effective in reducing distress. And I think that the important finding in this experiment was the effect of empathy on the effectiveness of touch. In this correlation analysis, we found that the level of partners' empathy significantly predicted the level of pain experienced by the target. So basically, we found that only in the touch condition, the level of partner's empathy as measured by a trait empathy scale significantly predicted the level of pain of the target, which basically suggests that partners with higher level of empathy are better at consoling their partner pain. In a subsequent study, we wanted to examine the neural basis, the neural mechanism underlying this effect with a dual EEG system. So we used, again, romantic couples, and we had them coming to the lab, and they were connected to a dual EEG system. And we had similar condition of pain, no pain, touch, no touch. And we focused on the alpha mu band, since previous study have been shown that the alpha mu band is a biomarker of the mirror neuron system or the observation execution system. So we extracted alpha mu band from each participant and looked for brain-to-brain coupling in the alpha band. Okay, so we basically measured alpha band from each participant in this 4 condition, and we looked for level of coupling or brain-to-brain synchrony during the different conditions. So in this figure, we can see the level of the number of significant correlation that we had in different electrodes in the between the dyads of the participant. Each yellow line here represents a significant correlation after correcting for multiple comparisons. And so we have here the partner no touch, no pain condition, partner touch, no pain condition, partner no touch, pain condition, and the most important condition, this is the consolation condition, where the target experience pain and the observer or the partner was holding their hand. And we see that the highest number of links, the higher level of brain-to-brain synchrony was observed in the consolation condition, suggesting that during consolation, we see higher level of brain-to-brain synchrony compared to the 3 other control conditions where there was no touch or no pain. So this basically suggests that there is increased brain-to-brain coupling during consolation. And importantly, we found that this brain-to-brain coupling also predicts the level of pain reduction. In this figure on the left, on the bottom, we can see that the level of brain-to-brain synchrony significantly predicted the level of pain experienced by the target, suggesting that higher level of synchrony predicts lower level of pain. This basically suggests that perhaps when we see higher level of brain-to-brain synchrony, there is perhaps blurring between self-other distinction, there is more sharing of pain, and the outcome is lower level of pain experienced by the target. We also found that the higher level of synchrony also predicted the level of empathy experienced by the target. Here it was situational level of empathy. We asked the observer to tell them to report how much pain they think that the target experienced, and we correlated the level of the pain the target experienced and the level of pain the observer believed that the target experienced, and we found that those that were more accurate at rating the explaining or understanding the level of pain of the target showed higher level of brain-to-brain synchrony, suggesting that indeed brain-to-brain synchrony represents some form of empathy, and it perhaps contributes to pain reduction during hand-holding. In a subsequent experiment, we sought to further examine this effect with Fmri to examine the neural anatomical basis of this effect. And in this experiment carried out by Adi Koretsky, we again focused on examining romantic couples. We had 20 romantic couples coming to the Fmri lab. They were randomly divided into target or empathizer, and the target was inflicted with pain using a thermode attached to the leg. And we had a hand-holding condition and a control condition involving holding a rubber ball. So it was a 2x2 design, pain, no pain, hand-holding, holding a rubber ball. Now, in order to obtain scans of both the target and the empathizer, we used the serial Fmri design. So we basically carried out the experiment twice. Once we had the empathizer inside the scanner and the target was outside the scanner. And in the second phase, the target was inside the scanner and the empathizer was inside the scanner. So although we did not scan them simultaneously, we obtained scans of both the empathizer and the target during the hand-holding and holding a rubber ball condition. So first we see that we replicated the behavioral effect that we found in the EEG experiment. We see that hand-holding significantly reduced level of pain. When we looked at the brain activation of both the target and the empathizer during the hand-holding, during the consolation condition involving hand-holding plus pain, we found remarkable shared activation in the inferior parietal lobe, the IPL, in both the empathizer and the target. We see in red activation in the IPL of the target during consolation, and in the empathizer we see a very similar activation in the same region during consolation. And when we extracted beta values from this region, we found significant correlation between the activation in the IPL of both the target and the empathizer during consolation, suggesting that some sort of synchrony or coupling between the brain occurred during the act of consolation. And we were very happy about this activation in the IPL because this is part of the mirror neuron system, part of the observation execution system that we frequently see activated during social alignment together with the reward system and the misalignment distress system. We also carried out a PPI analysis, a psychophysiological interaction analysis that allows identifying a region that were coupled in their activity with the IPL during consolation. And we found that the IPL activity was positively coupled with the superior frontal gyrus, a region that is frequently found in studies on emotion regulation. So we concluded that perhaps this coupling in the IPL between the empathizer and the target allows increased ability for emotion regulation in the target. So perhaps what happens during consolation, we see that some sort of coupling in emotional sharing occurs between the empathizer and the target. And this sharing allows activation or increased activation of frontal regions that are responsible for emotion regulation, and this increase the ability of the target to regulate their emotion and diminish the level of distress during consolation. So this set of experiments so far basically focused on physical pain. And the question remains is whether the same effect can be generalized to other contexts and other situations like emotional pain. What about emotional pain? Can we generalize this model to situation involving distress like sadness or anger? Another question is what happened when the interaction partner are strangers because or friends, because our experiment mostly focused on romantic couples. And also another question is about other emotional responses or empathic responses. In the previous experiment, we focused on social touch or on consolation. And in real life situation, we have a variety of strategies to reduce other people distress. We use facial expression, we use verbal responses. So we wanted to further examine this effect, and we developed a new paradigm that we call the emotional sharing paradigm. And in this paradigm, we have a condition where we ask two participants, students, strangers that come to the lab to verbally share with an observer or an empathizer a real-life distressful emotional event that occurred to them in the last year. So for 5 minutes, they are sharing an emotional event that occurred to them, and the regulator or the empathizer is listening and asking questions. And in a control condition, we ask them to just share with the empathizer a real-life neutral event, like telling them how did they get today to the university. And we measure the level of negative and positive affect that they experience before and after the emotional sharing. And we use Fmri to measure their brain activity during the emotional sharing. And we looked for brain-to-brain coupling in the inferior frontal gyrus and IPL, which are regions that are part of the mirror neuron system. So initial finding, behavioral finding, we show that before and after sharing in the target, we see a decrease in negative emotions. So we see that when the target starts the emotional sharing, he has high negative affect, but after sharing, the level of distress reduces. On the other hand, we see that in the regulator or the empathizer, we see an opposite trend. The level of distress increases. So basically, we see this emotional convergence between them. So they start in a far or a gap between them, but then they converge to be in the same level of distress during this emotional sharing. We also looked at synchronization in their body movement using an algorithm that analyzes videos and basically examine the level of changes in pixel in the left and the right side of the screen. So this way it allows us to measure the level of movement synchrony during the emotional sharing. And we found that there is higher level of synchrony in their body movement in the emotional sharing as compared to the control condition. And we are also looking at synchrony in their facial expression using a program that allows measuring facial expression. So we are also analyzing level of synchronization in facial expression, and we see high level of synchronization during facial expression during the emotional sharing. And also initial findings show that there is an increase of brain-to-brain coupling in the inferior frontal gyrus during the emotional sharing as compared to the control condition. So we see that emotional sharing is associated with increased brain-to-brain coupling in the inferior frontal gyrus, and we see also high level of synchrony in body movement. So if we started with this basic model of empathy where we see that the target experiences distress and it provokes an empathic reaction, emotional empathy, cognitive empathy, we see that when we look also at the behavioral manifestation of empathy, we see that it provokes some form of synchrony that also had a direct effect on reducing the distress of the target. So we propose that a more extended model of empathy that includes not only the immediate reaction of the empathizer, but also how the behavioral manifestation of the empathizer affect back the target. Now, another interesting issue is the issue of group behavior. As I mentioned in the beginning, empathy occurs not only in dyad, but also in groups and in real life interaction. Our interaction are not restricted to interaction with one participant. Sometimes we interact with groups. We see it across the animal kingdom in many species, in schools of fish, in flocks of birds, and in mammals. We see herding behaviors, and of course in human we see spontaneous forms of herding when we walk in synchrony, when we clap our hands in synchrony, and there are also forms of intentional synchrony, like when we dance or when soldiers are marching. So herding and synchronizing is extremely prevalent in day-to-day interaction. And we start to examine in synchronization in individuals with autism spectrum disorder, and it is already well known that individuals with ASD suffer from difficulties in imitation and coordination of behavior, both in motor coordination, also in verbal coordination. But the question remains is whether they also experience difficulty in coordination in a group setting. So we designed a task of motor coordination using a computerized task involving movement of a circle on a screen. So we had 4 participants coming to the lab. In the control condition, each participant is represented by a different colorful circle. In the controlled condition, they are just asked to move freely on the screen. So we see that they are just moving randomly on the screen. And in the synchrony condition, they are instructed to move together in synchrony. So we see that when they are instructed to move together, they become like school of fish, they move together in groups. So we recruited participant students 136 students, and they were randomly divided into groups of 4 participants. And then we divided them, we used the AQ scale to measure the level of autistic traits of each participant, and they were divided into low autistic trait group versus high autistic trait groups. And we used 3 measures of herding, a measure that examine directional synchrony, how much they change their angle of their movement, a level of another measure of cohesion, that measure how much distance they keep from the other participant, and separation, how much they separate between participants. And we found that, of course, there was a highest level of synchrony in the intentional synchrony condition as compared to the spontaneous synchrony condition, where they moved randomly on the screen. But we found a higher level of synchrony in the spontaneous synchrony condition as compared to a random group when we reshuffled all the groups. And so basically we see that there is spontaneous synchrony when we just ask participants to move randomly on the screen. When we divided them into low AQ, individuals with low autistic trait versus high autistic trait, we found that across 3 measures of herding, the directional synchrony, the cohesion, and the separation, the low autistic traits group performed much better as compared to the high autistic quotient group. And this was specifically evident in the spontaneous condition as compared to the intentional herding condition. So we concluded that individuals with high autistic traits are perhaps less synchronized. They have some difficulties in synchronizing with group members. They have some problems in synchronizing in the context of group behavior. And but this ability was a bit better in the instructed synchrony condition, suggesting that when they are explicitly told to synchronize, they can perhaps compensate and perform in a good manner. We also had in another experiment condition where we tested the synchrony in dyads, and we measured brain-to-brain synchrony in the IFG using the same task. So we asked participants to move in synchrony with the circle using this computer game, and we use Fmri, and we found increased brain-to-brain synchrony in the IFG in the condition where they were instructed to move together in synchronies, compared to a control condition where they moved randomly on the screen and the other circle was controlled by the computer. We further wanted to examine whether brain-to-brain synchrony may be affected by the context of group membership. So we examined differences in brain synchrony between homogeneous groups of participants, where both were Israelis, versus heterogeneous groups where we had mixed group of Israeli and Palestinian dyads. So we compared the behavior and brain-to-brain coupling of dyads involving either Israeli-Israeli or Israeli-Palestinian interacting. And we can see here that in the control condition, there was no differences between them. But in the mixed group, we see that the in-group dyads showed an improved performance in the third block. And in the intergroup dyad, we did not see this increase in performance in the task. The same trend was observed in the brain-to-brain synchrony in the IFG. Again, we focused on inferior frontal gyrus. We see that there was no difference in brain-to-brain coupling in the intergroup dyad in the control condition, But in the synchrony condition, we see an improvement or increase in brain-to-brain synchrony in in-group dyad, but there was no increase in brain-to-brain synchrony in intergroup dyad, in the mixed dyad. So, and this basically suggests that the context is extremely important also for brain-to-brain synchrony, and that it's easier for people to synchronize with their in-group as opposed to with an out-group member. And the final experiment that I will present today focused on triadic interaction in a family. And in this experiment, we focused on group synchrony in triads involving a mother, father, and a child diagnosed with the ASD. And in this experiment, we wanted to examine the ability of individuals diagnosed with ASD children at the age of 4, their ability to synchronize their movement with their parents. We also measured of theory of mind and motor sensitivity in a free play paradigm. And to examine movement synchrony, we used the paradigm of drumming, task of drumming. So basically we asked each participant was given an iPad, and they were asked to drum on the iPad. We had 3 conditions: a spontaneous condition where we just asked them to drum on the iPad spontaneously. In the next stage, they were asked to drum in a synchronized manner. They were asked to drum together. And in the last condition, the parents were instructed to follow their child when they drum on the iPad. And we basically measured the number of coordinated presses on the iPad to measure the level of synchrony between the parents and the child. So we found first that the level of synchronized drumming was the highest when the parents were following the child, but also it was quite high when they were instructed to synchronize, and it was the lowest when they were just drumming randomly. When we examined the different dyads that comprise the triads, when we looked at the mother-child dyad, father-child dyad, and mother-father dyad, we found that the highest level of synchronized drumming was in the mother-father dyad, suggesting that they had some difficulty in synchronizing with the child. And finally, we found that the level of mother sensitivity significantly predicted the level of triadic synchrony, suggesting that mother with higher level of synchrony, mother with higher level of sensitivity, perhaps contribute to higher level of triadic or familial synchrony in the task. So this experiment basically suggested it is also very important to look at the family and to look at synchrony within the family. And we are also now looking at the brain-to-brain synchrony and triadic interaction in the family. Now, for future direction, we are now extending these initial efforts to examine empathy as a dynamic behavior using learning empathy paradigm where participants learn to adapt their response based on feedback. So we are using a task of reinforcement learning to see how we can perhaps have paradigms that involve feedback and see if there are some participants that are better at learning how to change their behavior based on feedback. And finally, I would like to advocate in favor of those real-life paradigms that involve portable system and real-life ecological interaction that allows simultaneously examining behavior in dyads and group and triads. And this allows drawing a conclusion on social behavior that are much more related to real-life social interaction. And finally, I would like to thank my team, my students, my collaborators, and the funding agencies. And thank you for listening.
Really, really wonderful talk. Thank you so much. And at this point, it would be great to field questions. Yeah.
Yes, Shlomit.
So first of all, thank you very much. It's a very thought-provoking and very rich presentation. And if I may, I would like to hear your feedback to a slightly different framing that might go through all the experiments that you suggested. And I would like to introduce two words that we are all psychologically familiar with: agency and trust. And if we go even from the first experiment of the quality of touches, the only one where the one who is touched is also touching, in other words, has some agency, and in loss of control is the hand holding. In the other two, the touch doesn't have any control. Now, touching is a very, very sensitive thing because it's also possibly the greatest source of danger. And so touch really has those two aspects, that when we lose agency, touch can become very, very threatening. And this connects to this experimental notion of membership, to the way that I see it is more a matter of trust. Do I trust this person? Usually membership controls to that. The Israeli-Palestinian experiment is really go even to more complexities. I want to bring just one thing about the agency and the analgetic effect from swearing, which is the area which I'm studying. And there are a few studies from, I wrote down the name of the university, from Keele University in the United Kingdom, showing that swearing has analgesic effect. So this is really a situation where, again, thermal pain. I think, I don't know about other pains, but you are also tried it with thermal pain. And there seemed to be significant effect of swearing. So I think that the agency part of this whole empathetic network, I thought might be considered.
When you will think about empathy, you think about the agency of the toucher or the agency of the target?
The toucher has agency. The touched one. The
touched one. So the target, the one who's suffering, he's also an agent. Yeah, I think that this is true, but also this is perhaps why we see the most pronounced effect of analgesic effect of touch in the hand-holding condition, because hand-holding is more equal. We see in comparison to stroking, where one is touching and the other is rather passive. In hand-holding, it's more equal, and perhaps the target is also an agent in this in carrying out this effect. When you mention trust, I think that trust perhaps has a role here. But we found that the effect of empathy predicting and the effectiveness of hand-holding, this correlation was only evident during hand-holding, suggesting that only when there is really hand-holding, the empathy of the toucher affect the target. So something beyond trust, beyond what I believe and beyond what I feel about the target, something happens during this touch. Perhaps there is some communication of emotion that contribute to this effect of analgesia.
Thank you.
Okay. Yes, Franz?
Yeah, it's a wonderful presentation, and I've always wondered why people would test empathy in single individuals, because it's of course an inter-individual phenomenon. Your study with the hand holding made me think of Jim Cohn. You probably know his work, no?
Yeah, of course.
He did early stuff on that. I have a question. Since you test romantic couples, you have males and females.
Yeah.
So the obvious question is, did you find differences between the sexes?
Actually, we in the Fmri experiment, we examined sex differences, and we did not find any sex differences. We found equal effect of hand holding of males and females. So there was no sex differences.
And is that also what Jim Cohn found in his studies? Was it similar?
I think it also did not find any sex differences.
Okay.
Yeah. But he also found an analgesic effect of touch, and he found also a reduced activity in frontal region that we did not find in our study. So we had another explanation about this finding.
Anyway, yeah, it's a wonderful paradigm that you're developing. I like it.
Thank you. Thank you.
Hi, thanks so much for your talk. I thought it was really interesting. I wanted to ask if in the experiment where you were looking at kind of the emotional interactions that were more informal, if you looked at a relationship between synchrony and accuracy, and if you would expect that, or if you think that's kind of a distinct process, maybe more in line with cognitive empathy, whereas you were looking at more emotional synchrony.
So you ask whether we see that synchrony correlate with accuracy of emotion, of understanding the emotion. Yeah, we did not examine this, but it's a wonderful idea to look into it. So, so would you predict that there would be some correlation between them?
I would think potentially, but maybe that would differ for friends and strangers.
Yeah. Yeah.
Thank you.
Thank you. You did? Oh.
Sorry.
Hello.
No.
I'm Judith Jung. I'm very excited about finding out about your research. I'm a psychoanalyst and a contemporary psychoanalyst, and we have moved away from the explicit language of communication to the somatic expression of communication, actually. And your research and the psychoanalytic research about the nature of attunement, the nonverbal, pre-verbal nature of attunement is, of course, right now very central in psychoanalysis. But this actual shift into a physical touch is what is still very difficult when we are in the therapeutic situation. And your research, of course, beautifully supported the importance of empathic identification and attunement in the therapy as part of the therapeutic healing process. But the issue is, how do we reach and communicate a nonverbal connection and the role of touch? So in all of the attachment and child development, this is becoming very important. And my question to you is struggle with addiction, because there's very pre-verbal early childhood attachment trauma and a lack of compassion that's operative often in these psychological practical.
I can, okay. I couldn't, can you repeat the last sentence? I couldn't hear you.
How can you suggest that we can actually use or integrate touch consciously on the side of the therapist to treatment of patients that have very early attachment and attunement trauma?
Yeah, that's a very good question. The problem with studying touch is that it's only acceptable between partners and friends, and not in the context of psychotherapy or between strangers. And this is why we started to examine synchrony in the context of stranger and look at other behavior that trigger synchrony. So we are now looking at behavioral synchrony or body movement synchrony during interaction and during conversation. And actually, we also have another study now going on where we studied brain-to-brain synchrony in psychotherapy. We have psychotherapists and clients coming to the lab, and we measure the level of brain-to-brain synchrony during psychotherapeutic sessions. And we looked at, we are also interested in looking at how this synchrony developed during the progress of one session and between sessions. And we basically believe that the level of brain-to-brain synchrony perhaps also represent the therapeutical alliance between the psychotherapist and the client. And it has been shown before that the level of synchrony may perhaps predict the outcome of psychotherapy. So perhaps touch is a bit problematic in the context of psychotherapy, but other physical behaviors like movement and facial expression synchrony can perhaps enhance the level of synchrony in psychotherapy and perhaps increase the alliance and provide better outcome to the client.
Thank you.
Thank you.
Hi, Lynn had to go, so I've sort of taken over moderating here. I see that Jim Rilling has his hand raised.
Hi,
Hi.
yeah, I just wanted to say how impressed I was with the range of different methods that you brought to bear on this question. But your research made me think of some of the earlier work that Tanya Singer did on empathy for pain.
Yeah.
And I seem to remember her implicating regions more like the anterior cingulate cortex and the anterior insula as being involved in empathy for pain. But I think your work was implicating more the mirror neuron system and the inferior parietal cortex. So I was just curious if you had any thoughts about why your results seem to differ a little bit.
Yeah, well, basically in the Fmri experiment, when we looked at the contrast of empathy for pain versus no pain, we did find the activation in the anterior cingulate and the insula. We found activation in the IPL only when we compared the consolation condition with the other three control conditions, with the no pain condition or no hand holding condition. So I think that we found mainly activation in this region because perhaps during this consolation condition, there is some sort of emotional sharing that involves not only sharing of distress, but also some adaptation of the situation. The observer is adapting and synchronizing with the target. And perhaps this activation in this region perhaps represents this tendency to adapt to each other and this mutual alignment.
Thank you. Thank you very much.
Thank you.
All right, I'm not seeing any hands raised here. Well, there was. There was a question from the chat from Leah K. Are you there?
Hey, sure. Yeah. I'll just ask with my larynx. Thank you. Great talk. I was wondering about parental diagnostic status and how that's handled as a variable in those parent-child experiments, and how you might like to see it handled as a variable in the future.
Do you ask about the diagnostic of the parents with in the experiment with the ASD participants?
Yeah, like I'm super curious about what you see when the parents and the child share a diagnosis, or when,
Yeah, well, in these triads, we did not have parents that were also diagnosed with autism. So we only measured the level of mother sensitivity in a paradigm where there is a free play with the mother and the child. And then we asked the mother to watch a video of her playing with the child and explain their behavior and show some insight about his behavior. And we found that the level of insight and their sensitivity predicted the level of synchrony between them. But none of the parents were diagnosed with ASD. Only the child were diagnosed with ASD.
Cool, thanks.
Thank you.
Thank you. And Charlie Lane, you had your hand up at some point, but did you still have a question?
Yeah, thank you. I know we're kind of running up on the hour here. Kind of more about the consolation intervention and just kind of how that may be related to this notion of compassion and maybe the overlap of the empathy versus compassion that's in the literature, and any thoughts you have related to that.
Yeah, well, in the literature, compassion is associated with more rewarding and warmth feeling toward the suffering of the target. And I think that there is some overlap between consolation, empathy, and compassion. So in our experience, we did not ask about our participant about their how much compassion they felt toward the target, but it's possible that that they did experience some form of compassion toward the target when they held their hand. I think that in compassion, we see more activation in the reward system. Usually in experiment on compassion, we see activation in the reward system. And in our experiment, we did not observe any activation in the reward system. So I believe that even if there was some bit of compassion, it was not, they were mainly experiencing empathy, and they were sharing the distress of the target. So we found more activation in those observation execution region that perhaps represent their tendency to align with one another.
Yeah, thank you for that.
Thank you.
Makes me wonder, like most of the empathy experiments that we do actually tap into this idea of someone who is suffering and empathizing with that pain. Meanwhile, we could also empathize with someone's joy or happiness.
Yeah, that's true. We had in the past an experiment where we examined empathy for joy, and we found that the activation was in very similar region to empathy for distress, but there were lower level of activation. So I think that we experience mostly empathy for distress because empathy perhaps has evolved as a behavior that triggers prosocial behavior and helping behavior. And when we see someone in distress, it triggers some form of activity of us wanting them to help them. And if they are perhaps experiencing joy, they don't really need our help. So perhaps this is why sharing joy is perhaps less common and perhaps produces less activation in empathy regions.
Yeah, thank you.
Thank you.
Great. Well, thank you very much. Yeah, we've gone over the hour. Unless there are any final burning questions, maybe we can thank Dr. Shamay-Tsoory for a wonderful lecture and for entertaining our questions.
It was a pleasure. Thank you.